Thanks, Nina, for introducing me. I'm really excited to be talking here a little bit about how I got started working in drug discovery, chemistry, and AI. Just to give a little bit more of an introduction about myself, I'm 17, I'm in grade 11, and right now I'm working on researching basically applications for drug discovery using machine learning as an ML research intern at the Matter Lab, which is at the University of Toronto. So in addition to that, I'm also on the open source developer team for DeepChem, which is this project where we're trying to build a framework for basically understanding chemistry using machine learning, and it's based out of Stanford. So one paper we're actually working on right now is applying natural language processing techniques to understanding chemistry. And in the past, I worked at Integrate AI, which is this software company where I was building AI products for Fortune 500 companies. So yeah, I had a pretty long path to basically working here in a lot of different fields. So to get started, before high school, I was actually really interested in genome biology and computer science. So I love building random stuff with hardware, programming, running small bio experiments. But I didn't really have the belief or confidence that I could work towards solving a major problem and actually make an impact on it. But in my freshman year, I started getting a lot more interested in programming, and I went through this massive paradigm shift. So while I still had this same love for bio research, I took programming a lot more seriously. And then in October of 2018, I listened to this talk that really changed my life. And it was by Brendan Frey, who's this professor at UFD who's working on applying AI to discovering oligonucleotide gene therapies. And I never really thought I could make this kind of an impact on healthcare without being a biomedical researcher or like a clinician. But seeing how computer science is really being applied to some of the world's toughest rare genetic diseases and actually yielding drugs that were going into clinical trials was a really mind-blowing experience for me. So I was really hooked. But yeah, since then I kept learning about computational biology and machine learning until I reached the point where I felt comfortable enough experimenting and doing research on my own. And so with the help of some researchers at Deep Genomics, I spent nearly a year actually focusing on this area of applying machine learning to gene regulation. And the problem I wanted to solve was understanding DNA-protein interactions. So if you look at basically the drug discovery industry today, on average, it takes over $2 billion to develop a drug and 12 years to get from discovering a target to pushing out a drug with FDA approval. And so every year we're actually seeing an 8.4% per year decrease in new drugs per $1 billion invested into R&D. And so really the reason behind this is because biology is incredibly complex, because our human genome is made out of 3 billion A, C's, G's, and T's, which code for our entire life, but it's not really fully mapped out yet. One promising, actually, new development that's really kind of changing this is, despite this rising cost of drugs for genetic disease, we're actually seeing many tools which have been recently being discovered. One big example is sequencing. So because of gene sequencing, we're actually having genomic data becoming more accessible now than ever before. So we're seeing a 2.2x growth yearly in data sets. So when we first sequenced the human genome, it cost nearly $100 million to sequence a genome in the early 2000s. And now that's only $47. So really, to summarize, we have the tools to really modify our genome with CRISPR-Cas9 and gene editing. And we have the ability to generate vast amount of data by sequencing our genome at a really, really low cost. But we can't really use these tools to their full potential because we struggle to really interpret and understand how our genome truly works. So the big punchline here is that the genome is incredibly hard to interpret. If you take a step back and go back to its essential dogma, biology, which many of you learn in your introductory biology classes, we know that DNA gets transcribed and forms RNA, which gets translated and forms protein structures. But what ends up happening when we have these DNA mutations is pretty interesting. We basically get altered protein sequences because with altered DNA, it then eventually gets transcribed into RNA, and then it gets translated into a protein which is altered and doesn't actually carry out the same function. So these proteins are really essential for carrying out the functions that allow us to survive and thrive. So a lot of rare gene diseases are just as a result of improper gene regulation. And since we have all this data with the rising amount of genomic data, and we have really, really powerful computing using cloud computing, I sort of began to believe that we could actually use machine learning to help increase our understanding of the human genome. And so the way I do this is I'm actually using a common tool in computer vision, which is convolutional neural network based approaches. So if you look at images are really just these wide arrays of pixels, and we just take averages of these pixels and then make predictions off of them. For example, whether the image is a dog or a cat. But what we do is we literally take genomic data represented as an image through a process called one-hot encoding, and then apply literally the same approach to it, and we yield some pretty interesting results. So by doing this, we're able to predict the effects of genetic variants. So let's say I have a non-mutated gene sequence, the one on the above right here. I feed this into my deep learning system, I generate a score. That score is between 0 and 1, and it tells me how likely it is that a given protein will bind to a DNA sequence. Now I take another sequence. Let's assume that I know that there's a mutation, a C to G mutation. I feed that same sequence into this deep learning system, and I generate a score. Now, if that score is less than the non-mutated score, what I get is I get a negative score. And this negative score clearly tells me that this mutation has a negative impact on DNA protein binding. And it could be a link to a genetic disease that I might want to explore. So using this, we create this huge, elaborate sort of plots on entire genes. This gene right here is a small subset of the cholesterol gene called LDR1. And using this gene, we actually get a lot of different correlations about what mutations across the genome actually result in decreased DNA protein binding. One example you might notice if you look across this gene is the impact of C mutations. Across this entire gene, we see that C mutations have a very strong negative impact on DNA protein binding. So as a genetic scientist, I might want to choose to explore that. So this sort of developed into a really interesting tool for doing gene regulation-based research. So after spending all that time working in machine learning and computational biology, I actually decided to do something rapidly outside of my comfort zone. So last year, I was actually an intern on the MLS machine learning science team at a company called Integrate AI. So they're a venture-backed enterprise software company that's building a consumer intelligence platform powered by machine learning for a lot of different Fortune 500 companies. And so many of you are asking for why would someone who's working in computational biology and biology research choose to work and spend their summer working on a problem that's completely different. But I think that there's a lot of benefits that you can take from working at a high growth, fast paced startup that really translate to any field you want to work in. So first of all, you learn a lot about the different engineering best practices, from dealing with Git to working on complex applied machine learning problems like data imbalance or Bayesian optimization. And so many of these skills, for me personally, were incredibly valuable when I wanted to solve problems in machine learning, chemistry, and drug discovery. And so the main takeaway I really got from this experience was how transferable programming, engineering, and machine learning was as a skill towards solving some of the biggest problems in drug discovery. I also got the chance to lead a team of 6 engineers and ML interns. I think some of them are actually in this picture here, to actually build and deploy an internal open source machine learning tool that would detect key issues across their data pipeline. And so this gave me a chance to learn a whole new suite of skills about how to design a product for a core user, get feedback from users, write scalable code, and give really good feedback. And so the biggest takeaway I really wanted to share from my summer there was basically from this awesome experience was really how to apply an engineering and problem-solving mindset to problems in biology. So when many of you guys are working on your research projects, instead of trying to perfect every single detail in a research project from start to end, my advice would be to try to build a project to its minimal viable state. So think about what your minimal viable goal is for this research project you're undertaking and get that project to there. From there, try to keep iterating and improving your project until you get it to where you want to be, and you'll likely move a lot more faster. And so the biggest piece of advice from that was really that I highly recommend that everyone really works towards trying to land an internship, because any actual real world experience in any field, whether it be working in a wet lab biology lab or doing full stack development, it's going to be much more valuable than actually researching it on your own, because you have mentors, you're working on real problems. So I highly recommend trying to find one if you can. So after spending that summer working on machine learning for business analytics, I went back towards working for machine learning and drug discovery. So I talked a little bit previously about how hard it is to basically find a drug, right? Drug discovery is now this billion-dollar, 10-year pilgrimage. So we have to take drugs through multiple clinical trials, multiple years of human and animal testing. And so we might go, for every 10,000 drugs that we test, we might only have one drug that actually successfully makes it to regulatory approval. So this is really, really expensive and just really, really a huge, painful task to take. But one thing that I think is really changing the landscape is really open source biology. And what I mean by open source biology is the emergence of these open source libraries like You might have heard of PyTorch or Autodoc. And really what we're creating as a result of these open source libraries, including DeepChem, is we're basically allowing anyone with a computer who actually has the right knowledge to generate value in the discovery of new drugs. That's basically what I started off of. So first I started with developing an initial hypothesis. My initial hypothesis for building a COVID antiviral was that we could use graph machine learning to filter and screen a bunch of repurposable data sets. So I reached out to a lot of researchers working on this problem at MIT, Stanford, the University of Toronto, and I got a lot of feedback on, does this solution have a chance at maybe solving this problem? Once I got a lot of feedback, I iterated on my hypothesis and I got to work. So I started building a lot of my computational models and started executing on my workflow. And now I'm actually working on writing a white paper for this project. And I hope to hopefully get into some sort of viral testing partnership for my small molecule drugs. On a high level, what a small molecule is and the hypothesis for my approach is you can think of a molecule as a key and a protein as a lock. So basically proteins have these small grooves which small molecules can bind to. So you can kind of think of it as a key fitting in a lock. You want to figure out how to get that key in the optimal 3D position to fit effectively into that lock. Once you get a small molecule drug that can successfully bind and fit into your protein, you can then suppress the function of a protein. And in the case of the protein I'm targeting, it's a protein called 3CL protease or main protease. And what it does is it's a huge contributor towards actually the virus's replication process. So if we can successfully design a drug that binds to this protein, we might be able to slow down the process of the virus's replication. And there's many existing drugs that are being tested for this, including lopinavir. So on a really high level, not to get too deep into what I'm doing, I'm basically using graph-based machine learning to convert molecules into complex graphs. And the reason I do this is it actually optimizes for a drug's chemistry. So after I did this, I actually screened over 30,000 molecules for their ability to bind and inhibit the 3CL protease. And I think this is really why, in a nutshell, why computational tools are so awesome and why I think it's a really good starting point if you're a high school researcher. Like for you to test 30,000 of these molecules in a lab would cost probably millions of dollars. And it's likely impossible even for some companies to do or even place a bet on. And so because you're able to computationally do it, while it might not be as accurate, it's a good starting base towards trying to solve a really tough problem like searching for a COVID small molecule drug. One of the biggest problems I actually ran into when I was doing this project was just the issue of lack of data. So many of you who have heard of machine learning, you know that it takes thousands of different data points for a model to successfully learn an actual patterns. But we don't have thousands of data points for COVID-19. But the good thing about that is COVID-19 actually has a big brother virus called SARS-CoV-1, which originated in 2003. And the real convenient thing is the protein structures in these two share a 96% structural similarity. So effectively, we have a lot of data for SARS-CoV-1. We did a lot of research. We tested hundreds of thousands of molecules on this protein target. So I can effectively train a machine learning model on a combination of both the SARS-CoV-2 data or the COVID-19 data I have and the SARS-CoV-1 data I have, and it generalizes decently well. So taking a look at the entire pipeline, I basically went from generating 24,000 molecules and using a data set of 6,000 molecules and screening it down to 10 molecules now that I'm basically looking to do in vitro testing on. And I'm currently organizing some viral testing partnerships to do that. So the last project I'd like to talk about really quickly, something that I've been working on in the last couple of months as well. Many of you have heard of natural language processing. It's what's behind Siri, Google Translate, or even autocomplete. You might have even heard of the company called OpenAI, and they're a company that's working on they created generative models that are able to create text based off any prompt that almost mirrors human written dialogue. And so since then, we've been able to create NLP that can create music, sheet music in the style of Beethoven, or rap in the style of Kanye West, or even understand the functionality of a protein sequence. So my idea is, what if we could use the capabilities of these really effective models called transformer models for deciphering and understanding complex molecules in chemistry? And so that's when I basically started this project called ChemBERT. And so what we're trying to do is we're trying to use a natural language processing model to use a string-based or text-based representation of a molecule. And using this, it's able to actually predict how likely it is that a given molecule will be toxic with over a 96% accuracy. So what's really cool about this is instead of having to test all these molecules in a lab, which again takes thousands of dollars per molecule, for whether it's toxic or not, a drug company could use these sort of computational models for doing the filtering on its own, and it saves a lot of time and money. Not only that, but we also have a model that's highly interpretable. So when I say interpretable, I mean a big issue we face in deep learning is we don't actually know what our models are doing half the time. So we're basically all just like trying different configurations and hoping it works and really crossing our hands like, oh, this will boost the accuracy of our model. But what's cool about transformer models is we can actually look at every single molecule and look at what substructures are most important in whether a molecule is toxic or not, or really what makes the molecule, gives the molecule its properties. So in this case, we actually visualize each of the atoms in a molecule and to tell us which ones are more important structurally for basically an entire structure of a molecule and which ones are less important. So techniques like this allow us to uncover key insights about what makes a molecule toxic. So not only are we creating models that are hopefully going to play a big part in the drug discovery process, but we're also creating molecules that can understand and actually help medicinal chemists understand chemistry. So right now we've written our first paper and we're looking towards finishing the paper in August for submitting to conferences. Yep, so my last slide, I just wanted to end off with some last pieces of advice for people looking to do research in high school, because I know it can seem pretty daunting, but I think these are 3 pieces of advice that if I had 2 years ago, I'd probably be even better off. And so the first piece of advice is really just to find a good mentor who's the best person in your field. So this is advice that I've gotten from a lot of mentors, and the reason why they say this is because, for example, at Integrate AI, I had a supervisor really push me to struggle at solving challenges that were way above my pay grade as like a lowly intern, but it really refined my skills as a data scientist, and the skills I learned from that really compounded. So the biggest thing I would say is, as an aspiring researcher, one of the best things you can do is find a mentor who's at a place you'd like to be 10 years from now. So think about, really set a goal for yourself. It might change. It's okay. We're all young. We can all change what we want to work on. But think about, as of right now, where would you like to be 10 years from now? And try to find a mentor who's done that, because they can really guide you through a lot of the mistakes they've made and help you avoid those mistakes. The second piece of advice is really just to avoid chasing titles or status, no matter how convincing it might be. A lot of you guys might have seen like other teenagers who speak at global conferences, who are TEDx speakers or are on covers of magazines for being young entrepreneurs. And I think that's great if you can do that, but you have to remember that it's actually really easy to come across as impressive after spending a couple of months researching a topic. A lot of adults who don't really know the topic you're working on will say you're impressive, but I think that you kind of have to treat public recognition as a dangerous tool because you can easily get complacent. Really try to spend more time like following what your genuine curiosity is, and you'll produce much better research than someone who's working in a field that's highly publicized or getting a lot of sort of so-called clout for what they do. So I think really I think it's a good resource to have because you build your network and everything, but you want to make sure you're not sort of obsessed and chasing it like you're on an infinite treadmill sort of chasing that. The last piece of advice I have is to always keep building and experimenting and try not to stay complacent in one field. The reason why I say that is really because a lot of us are really young, and maybe after a week of exploring, we'll find one topic we really, really enjoy. But if we choose to spend like maybe 10 years working on that topic, you don't know about the countless other topics you might have been interested in researching that you just decided not to do because you found something and got really comfortable working in it. So really my last piece of advice is to keep exploring, keep your curiosity, and you'll probably be much more likely to find the topic that you're truly going to be the greatest at researching. And yeah, that's my talk. Thanks so much for listening.